My name is José, and I have finished my PhD this past semester on May, and now I'm working on my post-doc with my advisor, Dr. Wade Thomason.
Hey, my name's Bermand. I am a year into my master's program, also working with Dr. Wade Thomason. I did my undergrad here at Virginia Tech.
So basically during my PhD project, I have been working with plant biostimulants. I start testing the products on corn, and I got good results in terms of biomass, and so in the greenhouse and outdoor studies.
My side of the research, I'm focusing on soil health and biological indicators of soil fertility. We're testing microbial products, so I'd like to see if they're actually having an effect on that nutrient cycling process. So I'm looking at respiration tests, which are known to be very well correlated with nitrogen release. I'm looking at protein availability and also the active carbon pool. So we're putting down microbial fertilizers as well as a very recalcitrant form of carbon, humic acid. So I'd like to see how that is actually playing out in that nutrient cycle. So we're really looking at the processes which those microbes are carrying out, not who they are. If there's a key microbe that's somehow magical, that's not really what we're looking at here. We're looking for the function and how they contribute to nitrogen availability. That's on the soil side, and like Jose said, we're also looking at how the plant is responding directly, because we're not only applying these fertilizers to the soil, we're also doing a foliar application that goes straight onto the leaves. So there may be some interesting things going on there with secondary metabolite production.
Biostimulants can be a very good alternative to boost productivity. So that's why we kind of merge with the hemp program, and we start testing this plant biostimulants on hemp. Basically, so that's pretty much the rationale behind this study. But our hypothesis is we think that we have found in some previous studies that humic acid can increase microbial activity. And our hypothesis that if you combine the application of humic acid and biofertilizers, the humic acid will increase microbial activity in the biofertilizers, and then will be beneficial like this synergetic effect will be beneficial in the overall outcome. So that's why we're basically testing biofertilizer alone, humic acid alone, and the combination of humic acid and biofertilizer. And then you see, like we got good results on in a hemp greenhouse study, and corn in the outdoor and greenhouse, and now we are testing the hemp outdoors, and we're pretty excited to see the results.
So we've got two different kinds of anaerobic biofertilizers that we're testing.
Right.
And then also the combination of those with the humic acid.
So one biofertilizer is actually fermentation process that takes 15 days to be ready without any extra oxygen in the system. It's just pretty much we put the manure in the tank with the water and some microbial food, and wait 15 days, the product get ready to be applied. The other biofertilizer is just a powder, dehydrated powder, with a bunch of microbials, beneficial microbials that are diluted in water, and so it's ready to apply. So no extra source of oxygen in the system. And the humic acid is a liquid form of humic acid, and so pretty simple. So I think basically that.
There are many claimed benefits from these products, such as the increased nutrient cycling, increased drought resistance, potentially increasing soil structure, maybe even reducing pest and disease pressure, but we really have chosen to focus on the key indicators of productivity and nutrient cycling. That's another thing that's kind of interesting about this year. We're working with a different soil type than what we did last year, so we can see if this maybe has a higher response to it.
Right, and also because maybe the main reason why we are just trying to keep it simple is because there are many potential benefits but we are trying to keep it simple to at least the key benefits to figure out if it's actually working.
Right.
So, and especially on hemp, which is it's a new, technically new crop. So we are taking it more we are taking it slowly to see the benefits and then go from there to the next step for the next study and stuff. So
yeah, so for our foliar application, we're using the backpack sprayer, I think at 30 psi, and we just walk over the rows and apply it as such with the boom. But we do the 2 biofertilizers individually, then we do the humic acid individually. Then in the in the main canister, we will mix half humic acid, half biofertilizer, and then do the opposite biofertilizer the same way and apply it to the plots as we have them arranged. So we're testing 6 different treatments, including the control.
29, 28, 19.
61.8.
64.4.
61.1.
60.5.
Some biostimulant products claim that they can affect flowering, and so we don't know yet because it's the 2nd year study.
And in future studies, we're hoping that we can dive deeper into the actual effect on the cannabinoid production.
Yeah, definitely.
We do think that these biofertilizers would have a pretty heavy mode of action there, especially just from what I saw with those real bright white trichomes. That's where the cannabinoids are stored, so if they're really producing a lot of those, they have a potential to be higher in CBD.